Welcome to week 4, students. Can you believe it? We are going to be halfway through the term at the end of this week. So let's see what we did last week and what we've got going on this week. So last week we looked at diffusion and osmosis, diffusion of that iodine solution into the potatoes, osmosis having to do with the dialysis tubing that acts like a cell membrane, simulates a cell membrane. You did an activity where you made cells and viruses and then you identified them through a dichotomous key. So you learned all about cells last week and also viruses. Viruses are not cells. This week we are going to look at cellular respiration and photosynthesis, which are two really big topics, but we will touch on them this week. Cellular respiration is the process where ATP is made from food that you consume as an organism and oxygen is used. Another name for this is aerobic respiration. Aerobic means with oxygen. Respiration, if you think about that, what is respiration to you and me? Well, when we breathe in, we take in air, which is consists of about 21% oxygen, and we use that oxygen to run all of our cells. And then when we breathe out, we're releasing carbon dioxide, which is a waste product. Lots and lots of organisms do cellular respiration. There are other ways to make ATP without oxygen, and these are anaerobic respiration and fermentation, and you will get into these if you are taking the lecture at the same time that you're taking this lab. But we will concentrate on cellular respiration in peas. Photosynthesis is a way that some organisms, the autotrophs on the planet, make their own food, and they make the food glucose by using sunlight as an energy source and carbon dioxide. So they take carbon dioxide and they latch these or bond these molecules together in a specific way and come up with the molecule glucose, C6H12O6. The waste product of photosynthesis is oxygen. So it looks like cellular respiration and photosynthesis are opposite reactions. And you can kind of think of them that way, but there's something else that you need to understand. The organisms that do photosynthesis also do cellular respiration. So they make their food glucose using sunlight and carbon dioxide. But they also then use that glucose in cellular respiration to make ATP. So that's very important. So just realize they are sort of opposite reactions, but the organisms that do photosynthesis also do cellular respiration. So they're really not opposite in that case. Okay, let's talk about the lab this week. I mentioned last week in the announcements that you're going to have to start this experiment at least 4 days ahead of time because you have to soak the peas in water, the peas that you're hoping are going to germinate, which they did for me, so I'm sure they will for you too. You soak the peas overnight. This is not going to take you long. One word of caution, make sure you use water that does not have chlorine in it. So use filtered water, use bottled water, use distilled water. You can even use tap water if you let it sit out in a container at least overnight, but preferably a day or two. Okay, you got to get the chlorine out because if you don't, the chlorine will shut down the peas and you won't get germination. The chlorine will kill the peas. So the first day, this has to be done at least by Thursday so that you can get finished with this experiment. Soak the peas overnight, as described in the lab instructions. The second day, put the peas on wet paper towels overnight. Don't let them dry out. If they dry out, they'll die. So you can't let them dry out. On the third day, you need to set up the respirometers. And these are the contraptions with the tubes and the pipettes that you're going to set up per the instructions in the lab, and then conduct the experiment. Potassium hydroxide is caustic. You need to be careful with it. You need to be safe. So follow all the safety measures. Wear your gloves, wear your goggles. Don't let anybody else in your room when you're doing this. Be careful with the waste cleanup. So just be cautious with this. The whole purpose of this experiment is if respiration occurs, then what are you going to make as a waste product of cellular respiration? Carbon dioxide. Well, potassium hydroxide reacts with carbon dioxide and forms a solid. And so this means that gas will be used up in the respirometer. And you're going to see the dye that you put at the end of the tip of the pipette move inward toward the tube that you have where the P's are located. So that's the whole premise behind this experiment. For me, I found that 20 minutes was too long because all my respiration happened within the first, say, 10 minutes. So you may find that too, but that's okay. Just see what you get and stop the experiment when you can't read one of the tubes any longer. I will also post a video that I've made on how to use parafilm because it tells you to use parafilm in the instructions, but of course you don't know how to use parafilm. You've never even seen it before. Parafilm is a stretchy wax paper, which is interesting. I wish they sold it for household use because it is so cool. It stretches, and it stretches over surfaces well, you're going to stretch it around the end of the pipette, but also the end of a tube and all sorts of things. There are all sorts of uses for parafilm, and I will put that video in the announcements also so that you have that as a reference point. Other than that, I'll be doing a bunch of grading this week from week 3. So be watching for that. You have your final project that you turned in first milestone for. I'll be grading those in the labs from last week. If you need me, here's my email address, m.sigmon@snhu.edu. Please email me if you need help. I will get back to you as fast as I can. Last week I was doing a bit of traveling, so it made it a little bit harder for me to get in touch with you very quickly. But this week I'm back to normal, so I'll be here and answer as quickly as I can. And I hope you have a wonderful week.